we were discussing innate immunity, and we had discussed the surface protective mechanisms with respect to different regions of the body, starting from skin, then the eye surface, gastrointestinal tract, the mammary gland, urinogenital tract. Now, the next region of the body which is exposed to the pathogens continuously is the respiratory tract, because we don't stop respiring. We respire all the time. Since there is respiration and there are microorganisms in the environment, they can invade the respiratory tract anytime. One thing you remember, all these organisms are opportunistic invaders. Opportunistic invaders means they take the opportunity of one's body immune system. When it is weak, they are capable of causing the infection and the disease. Otherwise, our innate mechanism most of the time sees to it that that infection does not take place. Now, first, let me take the nasal passage. Nasal passage does produce mucus, and that mucus is pushed towards your buccal cavity periodically by the ciliated epithelium. That is, you can call that as upper respiratory tract. The lower respiratory tract is starting from your trachea, which splits into two bronchi, and those bronchi enter the lung. Up to the tip of bronchi until they enter the lung, and the tracheal inside surface, tracheal epithelium and the bronchial epithelium. Both these are provided with ciliated epithelium. And that ciliated epithelium beats the cilia in the opposite direction. That is, not towards the lung, but towards upward direction. And the mucus secreting glands that are there in the epithelial lining, and that mucus is constantly being pushed upwards without your knowledge, and that mucus comes to the pharynx region and gets emptied into your digestive tract. And sometimes that mucus is very thick. It comes and settles in your throat, and you need to clear it. Yeah, you know that how you clear it. Once in a while, you may cough it out, or you have to just give just a push to it, so it will go into a digestive tract. So remember, this mucus that is secreted by the epithelium in the respiratory tract, that is in your nasal tract, that is nostrils, as well as the tracheal, as well as the bronchial epithelium, is responsible for secreting the bacteriostatic defensins, which we'll be discussing in few minutes from now. Bacteriostatic defensins, which are proteins, as well as secretory immunoglobulin A, IgA. Secretory immunoglobulin A makes its presence here also. I've given this example. In the gastrointestinal surface also you will have secretory IgA. In your saliva you have secretory IgA. In your tears you have secretory IgA. Remember that. Whenever IgA makes its presence in the secretory fluids, it is called as secretory IgA. Details about that when I'm going to talk about the properties of immunoglobulin A, I will discuss. Now, for the timing, you keep in mind, a non-specific IgA is also there, which is bacteriostatic. Defensins and IgA in the mucous surface of the bronchial epithelium protect your bronchial epithelium. The second function of the mucus, which is there in the upper, that is in your nostrils, as well as lower respiratory tract, is to trap the dust particles that enter the respiratory tract. The dust particles of size anything beyond 0.5 micrometers, those which are larger than 0.5 micrometers, are trapped on the mucous surface of your respiratory tract. That is in the nostril as well as in the trachea and the bronchi. And these particles that are trapped are pushed by the mucus into the pharynx and they enter the digestive tract. But any particle which is less than 0.5 micrometer in size cannot be trapped by the mucus. It will enter straight into the respiratory lung surface and damage the lung surface. That is why, those people who work in the industries which produces dust, they must protect their respiratory tract from the dust. For example, cement industries and rock crushing industry. That is the people who work for making the pellets, rock pellets in the quarry region. Those people have to protect their nostrils. Otherwise, the particles of less than zero point 0.5 micrometer will enter the lung and cause a disease called as silicosis. And this disease is an inflammatory disease, silicosis, because silica particles cannot be digested in the body. Neither your immune cells, like macrophages, which phagocytize these inert particles, will be able to digest them. Once the macrophage phagocytizes these foreign particles that enter the lung, those macrophages become useless and they settle down in the lung to cause dense patches in the lung, which is called as granuloma. And those dense patches will be having large number of silica particles in the cytoplasm of the macrophage after the formation of the phagosome, and these macrophages will be unable to do any more defense function in your body, and that will result in a kind of silicosis, because once they settle in the lung they become inflammatory because they have encountered a foreign particle, that is the dust particle, and they will be triggered to produce inflammatory cytokines and lung damage will take place. Okay, so as far as respiratory tract is concerned, it is well protected, and you know that route of exposure to coronavirus is through your respiratory tract as well as from the oral passage and probably from your eyes. Okay, probably from your eyes. And if these systems are working very well and your innate system is good, that can also defend coronavirus. Okay, but coronavirus is just like any other influenza virus, and if your innate immune system is good enough, it can fight that. Now let us come to the antimicrobial peptides, which I kept on telling about defenses. The antimicrobial peptides are nothing but small micropeptides. These are small proteins not exceeding more than 100 amino acids. Maximum is 100 to 108 amino acids, and some of them are very small, even having 29 amino acid residues, and some have 40, 60 like that. And these defenses, antimicrobial peptides, which are produced, are by the epithelial surfaces, as well as some of them are produced by the immune cells. Immune cells. One of the immune cell is neutrophil. Neutrophil is naturally there in our body as a WBC, white blood corpuscle. Okay. So the defensins, which are a type of proteins which can be classified into alpha and beta defensins. They are based on their structure. You have heard about the alpha helical structure and beta pleated structure in the protein. Based on that, these defensins are also classified. Now, human alpha defensins are of 6 types, and in short form, they are called as small h ADS, human alpha defensins. Small h ADS, A and D will be capital. First, that is 1 to 4 alpha defensins, human alpha defensins, are produced by the neutrophils, whereas the human alpha defensin 5 and 6 are produced by the epithelial surface. One of the epithelial surface is in the intestine. In the intestine, there are cells called as Paneth cells. And where these cells are, these Paneth cells are present in the Paneth cells are present in the crypts. Crypts are nothing but the crevices or the valleys in between the villi. The intestinal surface has finger-like projections called as villi. Now, in between the villi, now you can compare that to between the junction of your two fingers. At the bottom of the finger, you have that junction, and that part can be compared to the crypt, CRYPT, crypts. And these crypts contain Paneth cells. And these Paneth cells are modified epithelial cells, and they secrete human alpha defensin 5 and alpha defensin 6, and that is copiously poured into the mucosal surface of your epithelium of the intestine. That is how they protect. And human alpha defensin 1 and 1 to 4, 1, 2, 3, 4 are produced by the neutrophils, and neutrophils are found circulating in your blood. That means 1 to 4 human alpha defensins are freely present circulating in your blood, and they are in your blood plasma. Okay, then there is also the epithelium, but that is protecting the vaginal surface, which I had told you. The vaginal surface is protected by mucus as well as the defenses. Human alpha defensin 5 is also produced on the vaginal surface. Next comes human alpha defensin, that is, sorry, human beta defensin. The human beta defensin 1 is produced by the kidney epithelial cells as well as vaginal epithelial cells. So, beta defensin produced by the kidney epithelial cells protects your urinogenital tract in men, and in the case of female, it is protecting the vaginal surface. So, there is also human beta defensin 1 produced by the lung epithelium. Okay, lung epithelium as well as the tracheal and bronchial epithelium, and that tracheal and bronchial epithelium is protected by the human beta defensin 2. Okay, human beta defensin 1 is produced in the surface of kidney as well as vagina, whereas human beta defensin 2 is produced on the lung epithelial surface, pulmonary epithelial surface, and it protects your respiratory tract. Okay, and it is capable of causing lysis, bacteriolysis. It is capable of conjugating with a substance called as lactoferrin. I had mentioned about this substance when I dealt with human milk. Lactoferrin produced by the mammary glands, and that lactoferrin which is produced in the human milk, okay, is capable of trapping the iron and deprive the iron for the bacteria. So the human beta defensin 2 produced in the lung epithelial surface is capable of trapping the bacteria, as well as it is capable of killing the bacteria by depriving the iron by conjugating with lactoferrin. There are other antimicrobial substances, which are one example is cathalycidins, which belongs to the family of protein called as cathepsins. Cathepsins are a type of proteins which are also micropeptides, small peptides, okay, and they are capable of inhibiting gram-positive and gram-negative bacteria readily, and they are produced on the epithelial surfaces. Cathepsin, that is cathalycidins, are produced on the epithelial surfaces, and they are capable of inhibiting gram positive and gram negative bacteria. And sometimes they are also called as cathepsin L inhibitors. Cathepsin L inhibitors. That is the acronym for cathalycidins. Okay, and they are strongly active against Pseudomonas aeruginosa bacteria, which is a gram negative bacteria. Okay, and they also capable of acting synergistically with the presence of lactoferrins and the lysozymes produced by the other systems, even the epithelial surfaces. They do show synergistic action. Next, antimicrobial peptide is a protogrin. The protogrin is a peptide that is produced by the neutrophils once again. And this protogrin is capable of, in fact, causing the bacteriolysis. Okay, and it is having about 16 to 18 amino acid residues and having a molecular weight of approximately 2000. Okay, approximately 2000 is the molecular weight, and it is capable of causing the bacteriolysis, and it is capable of even acting on the acting on bacterial surfaces, and it is uh, capable of inhibiting both gram positive as well as gram negative bacteria. Then comes another compound, the micropeptide, that is called as granulysin. The name comes to that is because it is stored in the form of granules in the cytoplasm of the NK cells. NK cells, that is a short form for natural killer cells. We'll be talking about these natural killer cells in innate immunity in the later part. Natural killer cells. Similarly, there are also a population of gamma delta T cells. These gamma delta T cells and natural killer cells are innately capable of defending our system. More details about that I'll be talking about it in later part of the innate immunity. But for the time being, keep in mind that these cells have granules in their cytoplasm, and these granules are nothing but granzymes, enzymes, granular enzymes of lytic activity. They are called as granulysins. Lysins for lytic activity. Granular enzymes causing lytic activity are called as granulysins. Instantaneously, whenever the NK cells or gamma delta T cells, when they come across any bacteria in our body, they are capable of getting activated instantaneously and bring about killing of that bacteria. Okay, therefore natural killer cells get the name, they are naturally killers, therefore they are natural killer cells. Gamma delta T cells also killer cells. Okay, they are both capable of killing the bacteria instantaneously as they come in contact with the bacteria by producing those granulysins, and they make these granulysins enter the bacteria through a channel called as the the, the protogrin channels, and they also have the channels which are made up of tube-like structures which are called as the tubular channels, perforins. Okay, perforins, and these perforins will be inserted into the bacterial membrane, and these granulysins are emptied into the cytoplasm of bacteria, and with the result the bacteria is lysed. Okay, and once again they are also small peptides. Okay, granulysins. Then comes histatins. Histatins are derived from the amino acid histidine. Okay, histidine is a heterocyclic amino acid, and they are derived from that by modification in the saliva, and salivary enzymes are responsible for modification of histidine to histatins, and they are present in large amounts, capable of inhibiting not only bacteria, gram positive and gram negative bacteria, they are also capable of inhibiting some pathogenic fungi called as candida. Candida albicans. Candida albicans is a pathogenic yeast. Okay, it sometimes infests some people and it causes gum infection, and bleeding of the gums takes place because of the gum infection. And these people will have, because of these bacteriostatic agents are less in them, they may be having this problem. Therefore, histidines, which are secreted in the saliva, together with salivary enzymes, with our lytic enzymes, plus the bacteria, that is resident bacteria, resident flora of the mouth, which produces peroxidase, all this will protect your oral surface, that is the mouth region, that is buccal cavity. Then comes another peptide, which is about 108 amino acid residues, that is secretory leukoprotease. The secretory leukoprotease is the enzyme, is a enzyme produced by the epithelial linings of most of the protective areas. And this secretory leukoprotease is antibacterial as well as antifungal. And it is in fact capable of causing the lysis of the microorganisms. All right, and to some extent, some of the leukocytes, for example, the macrophages and neutrophils, in addition to the epithelial cells, also produce these leukoproteins. Okay, so therefore, the word first was coined because they were found produced by the macrophage and the neutrophils. They got the name leukoprotease because they are proteases, a type of enzyme capable of causing proteolysis. But they found that they are also present on the secretory surfaces of the epithelial cells. Therefore, they combined that leukoprotease with secretory and called them as secretory leukoprotease. Okay, and the word inhibitor comes because they are capable of causing all proteolytic digestions and inhibit the protein action. And proteins that are produced by the bacteria, which could be toxic, and some of them are toxins, and they are capable of digesting that protein by breaking down that protein. So therefore, these substances which are produced as antimicrobial substances on the surface protective mechanisms of our body help in giving us the protection through non-immunological means against the gram positive, gram negative bacteria, as well as the substances like the fungal substances, yeast, etc. Okay, so now we will move on to the other part of the innate immune mechanism. We have dealt with first line of defense. I have said that first line of defense is surface protective mechanism. It is all the mechanism that is there on the surfaces. Now the second line of defense is, suppose the first line of defense is breached, means the bacteria succeeded in invading the epithelial lining. It breached that epithelial lining and crossed that epithelial lining and entered your body fluid. If it enters the body fluid, your body fluid has different mechanisms of defending against the pathogen, and that is through physiological barriers. Okay, physiological barriers. Now let us take first example of innate immune response against the blood. Okay, our body defends entry of blood of any other blood group other than our own blood group. We tolerate transfusion of blood of our own blood group, but not any other blood group. That is an innate mechanism. And you know that it is always advisable to transfuse the blood of the same blood group into an individual to avoid immune complications. People might have said earlier that there is a universal donor and universal acceptor. That terminology does not hold good anymore as far as WHO is concerned. It is only in the case of emergency when the person has shortage of blood. At that time, you can use that principle of universal donor and universal acceptor. And only one unit of blood can be donated of the blood group O into other blood group people. A, B, and AB blood group people can receive the blood from blood group O person only once. Only once. Please remember that. If you transfuse more than 1 unit of blood, 1 unit of blood is equal to 250 ml of actual blood and 150 ml of the anticoagulant fluid with dextrose. And if you transfuse more than 1 unit of blood into any other blood group, that is O blood group, remember O blood group readily has antibody against A and antibody against B. A and B antibody is readily there in the O blood group person, and that antibody will destroy the blood corpuscles. And it is IgM antibody, and once it reacts on the RBCs of the A or B blood group, the RBCs will be lysed because IgM is a strong complement activator. So, blood will be hemolyzed if you transfuse more than 1 unit of blood of the opposite blood group, and the person will suffer from anemia and death. Therefore, please do not risk that principle of universal donor and universal recipient. There is no concept of that type. It is only an emergency concept, but nobody practices that nowadays. So please remember, our blood group is innately defending the transfusion of opposite blood group. I have to tell you some history behind this. Of course, you know that Landsteiner was the person who found the blood group. But before that, during Julius Caesar's time, during his atrocities against the people, to the criminals used to punish by transfusing the blood of animals. He would transfuse the blood of horse, he would transfuse the blood of dog, he would transfuse the blood of any other animal into a person who is supposed to be killed. And he did not know any physiology or medical science behind it. He knew that once you transfuse the blood of any animal into human being, the human being dies a painful death. And that is because of the innate mechanism that we have. And the criminals used to die instantaneously. And that time during his reign, it was allowed. And he practiced that transfusion of blood from animals into human beings to kill his enemies or to kill the criminals. That was his capital punishment. Now let us take another innate mechanism, that is the innate mechanism of physiological defense, that is called as alternate pathway of complement. I had mentioned that the complements are a type of proteins which are produced in our body, and they are responsible for getting rid of antigen antibody complex. And I had given this example when we did the test complement fixation test. More details about the complement, we'll be talking about that in unit 2 at the end of the chapter. But for the time being, keep in mind there are complements C1 to C9. Each has the name C1, C2, C3 like that up to C9. And all these act in tandem, one after the other. And they complement each other to get rid of antigen antibody complex. Now we will see how the complement helps in innate mechanism. Now we'll take the complement that is C3. Therefore, it is called as alternate pathway of complement. The normal pathway of complement is called as classical complement pathway. The classical complement pathway comes in the acquired immunity. Means once a person has developed immune response, when there is antigen antibody complex, to get rid of that antigen antibody complex, the classical complement pathway helps. Whereas alternate pathway of complement is innate. It does not need, or there is no necessity of antigen antibody complex in the blood for activation of this complement. The C3 complement has an innate ability of recognition of the bacteria. If the bacteria has breached the first line of defense and has entered the body fluid, this complement is always in circulation in our body. It is produced by the liver as well as the macrophages. And since it is in circulation in our body fluid, that is in the plasma, as soon as it comes in contact with the bacterial surface, that is bacterial membrane, it breaks down undergoing autolysis. It breaks down on its own. Autolysis means self-lysis, and breaks down into two components, that is C3b and C3a. C3b component, which is formed, will remain attached to the surface of the bacteria. That is called as surface bound. Bacterial membrane surface, it will attach and remain. Whereas C3a is released. C3a is going to cause anaphylaxis. We'll talk about that when we are going to talk about hypersensitivity and anaphylaxis. But keep in mind, C3a gets the name anaphylatoxin. It is capable of causing adverse reaction. It is called as anaphylatoxin. Let us not bother about C3a now. So C3 complement, on coming in contact with the bacterial surface membrane, it is split into two components, C3b and C3a, and C3b remains bound to the bacterial surface. And the C3b, which is bound to the bacterial surface, attracts a factor called as factor B. And that factor B is present in the serum. As soon as factor B attaches to that C3b complex, it becomes a substrate. It becomes a substrate for a proteolytic digestion by an enzyme, that is factor D. Factor D is an enzyme. It acts on the factor B and waits for the factor B to form a complex with C3b. You have a complex of C3b, capital B complex, to which factor D reacts and splits factor B into BB and BA. BA is released. Okay, BA is released, whereas C3b was already bound to the bacterial surface, to which now BB is complexed. BB is derived after the breakdown of capital B by the action of factor D. Once C3b BB complex is formed, it is stabilized by a protein, so stabilized by a component factor called as P. P for properdin. 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 Properdin is a factor. Properdin. Properdin. And that properdin stabilizes that factor C3b BB. And C3b BB, which is stabilized by the properdin on the bacterial surface, now denatures the bacterial cell membrane. And the denaturation process of the bacterial cell membrane is called as opsonization. OPSO, opso, NIZATION, opsonization. Opsonization means denaturation of the membrane of the bacteria. When the membrane of the bacteria is denatured by this complex, that is C3b, BB, stabilized by protein complex, that bacteria starves to death. The bacteria takes in nutrition through its plasma membrane by secreting the enzymes outside. It does exocytic digestion. Remember, it secretes all the enzymes extracellular and outside, and digested material is taken in by the membrane. Now, the bacteria will be incapable of sending the enzymes out for digestion nor taking the nutrition inside. It is starved to death. Okay. So, another very important point about C3b is that if C3b complex is formed and that C3b before joining B can be in fact binding to C3b receptor of the macrophage, that is an immune cell, which is innate immune cell. Macrophage can recognize C3b that is readily bound to the bacterial surface, and using the C3b receptor it can engulf the bacteria. And engulfing bacteria through receptor surface is called as endocytosis. So you know now the alternate pathway of complement, that is C3 complement, serves dual purpose in the innate immune mechanism. One, it is by itself capable of causing denaturation of the bacteria by forming a complex on the bacterial membrane surface as C3b-BB complex, which causes the denaturation of the bacterial membrane surface, which is opsonization, resulting in death of the bacteria. Second, by itself, when C3b is bound to the bacterial surface, it is recognized by the macrophage, and macrophage has C3b receptor. It comes and binds to that bacteria at the C3b position and engulfs the bacteria, and endocytosis of the bacteria results in killing of the bacteria in the macrophage cytoplasm. Now you move to the left hand side of the diagram. Right hand side of the diagram depicts the innate mechanism of protection against the bacteria. Now the host cells also can bind to C3b. Host cell is the person who is infected by the bacteria. His cell has to be protected because his cell also should not undergo opsonization. Remember that his cell membrane is also capable of undergoing opsonization if C3b goes and binds them. And how to protect the host cell surface? The host cell surface is naturally protected against the opsonization by the C3b complex formation by the production of a substance called as membrane cofactor of proteolysis. Membrane cofactor of proteolysis, which is called as MCP, membrane cofactor for proteolysis or of proteolysis, and decay accelerating factor. The decay accelerating factor. And what do these contain? They contain the factors called H and I. Factors H and I. And what does the decay accelerating factor do? The decay accelerating factor, that is H, is capable of splitting C3b when it forms a complex with B. Remember, C3b, when it binds to the host surface, host cell membrane surface, it can also bind to factor B. But the moment factor B comes and binds there, H factor, which is there in the host plasma, will split that factor. By splitting C3b from B, it will prevent that complex from forming on the membrane. Factor H is capable of splitting B, capital B from C3B. Now it should not allow that factor to recombine back with C3B, right? On the host cell surface, the factor I forms a complex with C3B to form C3BI. And when it forms a C3BI, it is inhibited from reforming the complex with capital B. That is how host cell surface is guarded. But this mechanism is present on only the host cell surface membrane. But this is not present on the bacterial cell surface. Unfortunately enough, if this mechanism, okay, is not there on the bacterial surface membrane, if it was there, bacteria also could have avoided this mechanism of its killing. This mechanism of not allowing building up of C3b complex on the membrane is present only on the host cell surface, but not on the bacterial surface. On the bacterial surface, that C3b B complex formation takes place successfully. Therefore, majority of gram positive and gram negative bacteria are tackled by this mechanism using alternate pathway of complement, and it does dual function. One, it is by itself capable of causing denaturation of the bacterial cell surface and killing the bacteria by starving. Second, it facilitates the macrophages to phagocytize the C3b complex formed bacteria, and after the endocytosis of the bacteria is going to be killed in the cytoplasm by various mechanisms. Those we will deal with in the next class. I hope you understood the alternate pathway of complement and other innate mechanisms that I explained to you about the antimicrobial peptides and etc.